Listen carefully. I'll be telling you some striking things here. Some organisms have nucleus. There's DNA in their nucleus. There are DNA in their mitochondria. While in plant, they have DNA both in nucleus, in mitochondria, and in chloroplast. But in RNA, it's distributed throughout the whole cell. There is not like RNA is in the nucleus, is in the mitochondria. No, RNA is distributed around the whole cell. And that is why disease condition affecting RNA is very dangerous, because it multiplies very fast. Genome organizations. Genome organization of an organism is a background layer of genetic information. When we say genome, you know, it is some total of DNA that is genome. So if DNA is a genetic information of an organism, means this genome comprises of all the information in an organism. So this genetic information provides multiple layers of information to the structural genome from the array of nucleotide sequence. This nucleotide sequence have all the information that is presented to direct the activities of the organism. And this is how it happens. The genome, which is the DNA, transcribed to transcriptome, and then the transcriptome translate to protome, that is protein. So the sum total of protein in the body is a protome. Genome organization differ from organism to organism, and the size and the activity increase with increasing complexity of organism. Means the more complex, like now we have unicellular organism, we have multicellular organism. So the number of DNA that is in unicellular organism will not be the same with the number of DNA in multicellular organism. So as the organism keep increasing, so also the number of the DNA and the activity also keep increasing. That is what we mean. And then I want you to know this, that we have earlier mentioned to you, I mentioned polynucleotides. And we mentioned array of a polynucleotide sequence, that is the genetic information directing the activities and the life of an organism. Okay, having established that, so for we to be able to have a nucleotide, we must have a pentose sugar. We must also have a phosphate bind to the pentose sugar and a nitrogenous base. But now we are starting with what? A pentose sugar. There are so many types of sugar. We have 6 carbon atoms, we have 4 carbon atoms, we can even have 6 and 7. But for the sake of this nucleotide, we are only dealing with what? 5 carbon atom, which is a pentose sugar. And the numbering is in prime. Look at it, we have 1 prime, 2 prime, 3 prime, 4 prime, and then the 5 prime. And then if you look at this place, 2 types of sugar that are present in nucleic acid. We have 2 types of sugar. One is the deoxyribose sugar and the ribose sugar. Deoxyribose sugar is found in DNA, while the ribose sugar is found in RNA. And if you look at this structure, you see that the thing look alike, but there are still difference. The difference is in carbon number 2. Everybody look at carbon number 2. This carbon number 2, you see differences. This one, it is only hydrogen attached here. Means the oxygen has been removed. That's why we call it deoxyribose. But here, our OH is attached here. That's why we call it what? The ribose sugar. Every other thing, they are what? The same. And then I want you to tell you a striking thing here, that the reactivity of this compound is not the same. Because this one, the oxygen has been removed, while this one still have its oxygen. So the reactivity, they are not the same. Now we are moving to nitrogenous bases. In nitrogenous bases, we call it their planar, the aromatic ring. Some of you know, like benzene chemistry. From your knowledge in chemistry, there's something we call benzene ring. This is one ring, this is another ring. 2 rings combining together, we call it bicyclic. So we call it their purine. And then look at this one, this is only 1 ring, we call it what? Pyrimidine. And then this is how the numbering is now. I want you to listen carefully. in this purine, why will you know it is purine? Because it is two rings combined together. That's why we call it purine. They have 9 members ring. 9 members ring. Look at it. 1, 2, 3, 4, 5, 6, 7, 8, and 9. That is what? 9-member ring. Then how many hydrogen do you have? We have 4 hydrogen. 1 hydrogen here, 2 hydrogen, 3 hydrogen, and 4 hydrogen. So it is 4 hydrogen we have here. So anytime you are giving out the structure of purine, the hydrogen inside must be 4. That is what we mean. Then how many carbon atoms? We have 5 carbon atoms. Look at the carbon atom. We have 1, 2, 3, 4, and what? And 5. So the carbon atom that is here is 5. So we have 4 nitrogen and 5 carbon atoms in purine. Now we move to pyrimidine. Pyrimidine, we have 6 members. What do we mean by 6 members? Look at the counting. Why this pyrimidine, they count it from side, but why this in purine they count it from nitrogen by the side? Why this one in pyrimidine they count it from the nitrogen at the bottom, which is 1, 2, 3, 4, and then 5 and 6. Then, and looking at it carefully, I want you to understand that we have 4 carbon atom, means the carbon atom in this pyrimidine is 4. Let's count it. 1, 2, 3, and 4. And then how many nitrogen do you have? You have 2 nitrogen, 1 and 2. Using the knowledge that we have already been established, now we are going to name our nitrogenous base. How many nitrogenous base we have? We have 5 nitrogenous base. And then number 1 of the nitrogenous base is cytosine, 2 thymine, 3 uracil, 4 adenine, and then 5 guanine. And then in pyrimidine, how many nitrogenous base we have in pyrimidine that are pyrimidine? Is 3 nitrogenous base that are pyrimidine. And these 3 nitrogenous base, this cytosine represented with letter C, thymine represented with letter T, and then uracil represented with letter U, adenine represented with letter A, and then guanine. So adenine and guanine, they are what? Purine bases. Component of nucleic acid. Now we have heard about nucleic acid. Nucleic acid are DNA and RNA. What are the components of nucleic acid? I want you to listen carefully. Under your DNA and RNA, there are nucleic acids that are both DNA and RNA. Means this nucleic acid, this is a nitrogenous base. You can see them in DNA. You can also see them in RNA. One of these nitrogenous bases is what? Adenine. 2 of these nitrogenous bases is what? Guanine. 3 is cytosine. So this adenine, guanine, and cytosine, it is found both in what? DNA and what? And in RNA. And thymine is only found in DNA. You can't see thymine in RNA. Why uracil is only found in RNA? You can't see uracil in DNA. Remember, we are talking about what? Component of nucleic acid. But apart from the nitrogenous base that I just mentioned, which are 5, we also have what? Our ribose sugar. In case you are not clear with this structure, in case this structure you are seeing, the way you are looking at this structure, it is not clear for you to grab. Now you can grab this one now. Look at how the structure is drawn. Look at it. There are so many structures here that can give you clarity. Look at the way it is drawn. This one is clearer for you to learn. At the position of the ribose, our OH here, our hydroxy group, they are complete both in 2 and in 3. And this one, we have only hydrogen attached. The oxygen has been removed, means there's a ribose. And then our phosphate group, with our phosphorus at the middle, binded to what? 3 hydroxy group, and then on top, with a carboxylic group of what? Binded. And this one now, we want to say here now is to establish for us how we can be able to have a combined structure that will give us a nucleotide structure. And one of these, look at our nitrogenous base here. At carbon number 2, it is binded to our ribose sugar at the carbon number 1. So carbon number 9 and 1, there is a binding there attaching to each other. Then at carbon number 5, we have it binded to what? Our phosphate group. So the complete of it will give you a nucleotide. Now, I want to tell you some differences here in this structure. You have heard of nucleotide, but there's something that I've not told you about. We call it nucleoside. And we say nucleoside is made up of 2 groups, sugar and nitrogenous base. Means in nucleoside there is no phosphate. In nucleoside there's no phosphate. While in nucleotide we have sugar, phosphate, and what? Our nitrogenous base.